Welcome to Hephzibah Simple Class. Today, I will be talking on transport of molecules across cell membranes. A cell membrane acts not only as a barrier, but also allows required substances to enter into a cell and cell products to leave the cell, as it controls the movement of substances into and out of the cell, while preventing entrance of harmful substances and exit of essential substances. Remember, this is made possible because of the selective permeability of the cell membranes. However, different processes occur in different parts of a cell. So, certain substances must be transported from one part of the cell to another. Therefore, if a membrane were to lose its selectivity, there could be a problem for the cell to maintain its internal environment. Transport of substances across a cell membrane is broadly classified into passive transport and active transport. A passive transport is the one in which certain substances like water molecule, oxygen, carbon dioxide, small carbohydrates, e.g. glucose, move across the membrane in their own natural tendency down the concentration gradient without an input of energy from this cell. A passive transport is subdivided into simple diffusion, facilitated diffusion, and osmosis. A simple diffusion is the movement of a substance down its concentration gradient. That is, the movement of a substance from a region of higher concentration to a region of lower concentration across a semi-permeable membrane. Osmosis is the diffusion of water molecule across a semi-permeable membrane. Facilitated diffusion is the diffusion of solutes through semi-permeable membrane, though with the help of special transport proteins like channel, carrier protein, or pump. Note that facilitated diffusion is specific. Movement of substances is down the concentration gradient, and it usually gets saturated once all the transport proteins are in use, which could cause no further increase in the rate of diffusion. What is active transport? Active transport is a type of transport which involves movement of substances like ions, large proteins, complex sugar, and cell across a membrane against their concentration gradients or against their natural tendency with the use of energy in form of ATP from this cell. An active transport is subdivided into endocytosis and exocytosis. Endocytosis is a transport in which a substance, be it solute or liquid, is captured from outside of the cell by engulfing it with its membrane and bringing it into the cell. Endocytosis is further subdivided into phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is a type of endocytosis which involves the capturing of solid substances outside a cell into the cell. Pinocytosis is a type of endocytosis which involves capturing of liquid substances outside a cell into the cell. Receptor-mediated endocytosis is a type of endocytosis in which specific substances recognized by membrane receptors are captured and transported into the intracellular environment. In conclusion, exocytosis is a type of vesicle transport that describes the process of vesicles fusing with the cell membrane and releasing out their contents to the outside of the cell or extracellular membrane. Here we come to the end of this class. I want to acknowledge and appreciate the authors and owners of every image and material used in this class. Sir and Ma, please do not forget to like, share, and subscribe to Hephzibah Simple Class YouTube channel. Thank you.